started and talk very slowly so people trickle in and won't disrupt anything. My name is Bryn Gardner, and I'm with the graduate student group Genomics from the Center for Ecology and Evolutionary Biology, and we are the ones that organize this for the evening. I wanted to take a moment and thank our sponsors before I get started with anything else. So we couldn't have done this without support from the University of Oregon College of Arts and Sciences, the government, VICE funds, the ACUO, the Center for Ecology and Evolutionary Biology, and especially tonight from the Oregon Community Center, so if we could thank them, I would appreciate it. So I was thinking about how to introduce our speaker and how to introduce the topics we're going to talk about today. And I thought the best thing I could do was do a 2-minute recap of what Bill Cresco talked about a month ago, which was broadly a brief history of life, and in particular, what makes us similar and what makes us unique. And what Bill talked mainly about was that it's both our experiences and our genes that make us similar to each other and different from each other. And then he went on to focus on particularly how our genes can distinguish us or make us similar, and the biology behind those genes. So I wanted to start with a really light topic, which is what is life. Turns out there's not actually a consensus for what life is, but biologists kind of agree on general things that can categorize something as alive versus not alive. So something that is alive can reproduce, can metabolize and grow, can maintain a steady state internal environment, meaning it's self-contained, and it responds to the external environment. The reason that we care about that is because life then can be thought of as things that go on in what Bill talked about as the central dogma of molecular biology, meaning that here at the top we have DNA, which is where all of the information about our life is stored. DNA can replicate itself, so here's this reproduction part. And then also, DNA, through an intermediate of RNA, makes proteins, and the proteins interacting with each other is what allow the rest of the life to happen. So it's proteins that are responsible for metabolism, for growing, for maintaining the steady state, and for responding to the environment. Of course, it's more complicated than that because proteins interact with RNA to do this, and DNA has a small part to play too. But we're going to call this the central dogma of molecular biology. One of the interesting things about the summary of life on Earth, and particularly with when we're talking about the central dogma, is that life is actually really old, but most of what we think about with life is not actually that old. So what do I mean by that? The Earth is about 4.5 billion years old, and first life, meaning unicellular organisms, appeared about 3.8 billion years ago. But it actually wasn't until, I have my notes here. It wasn't until 1 billion years ago that multicellular organisms evolved. And then it was only 200 million years ago that we got mammals. And then 65 million years ago is when dinosaurs went extinct. So we went from 5 billion years to 65 billion years ago. And then humans, the behavior of modern humans only appeared 200,000 years ago. But through all of this, we're still getting DNA replicating and DNA using RNA to make proteins. And so through all of this, we're actually getting DNA turning, so DNA starting from one parent and dividing, right, going into its offspring. And in this time period, we get mutations that occur. So in this star here, if this is going to be a mutation in one offspring, now all of the offspring that follow from this strand are also going to carry that mutation. And all of the offspring that follow from this strand are not going to carry that mutation. And then, you can imagine later on, just going farther down, there's going to be another mutation and another mutation. And so what that means is that we're actually going to be able to, through the entire history of life, trace these patterns of mutations and identify how we're all related to each other. And not just how humans are related to each other, but how all organisms are related to each other. So in this example, we're going to take a genomic perspective. So if we sequence the entire we can actually see how diverse life is on Earth. So this blue here is all prokaryotic organisms, single-celled, very, quote, basic. Here we have a different type of single-celled organism. And then if we want to wonder where humans are on this tree, where this tiny little branch right there, there's fruit flies, there's flowering plants. This is the rest of diversity of life. So I just wanted to use that to put everything in perspective for you. So that's what Bill talked about last month, and that's also a little bit about what Carlos Bustamante is going to talk about on May 23rd, how we can actually look at these patterns of genetic mutation over time in humans and identify how humans have migrated over time. We can do the same thing in canines and in different plants. We can use the same type of techniques. What Dr. Silver is going to talk about tonight is actually the second half of the central dogma. So here we have DNA. Here we have RNA. RNA helps make proteins. Proteins do all of this work. Well, I said before that we get mutations that happen in every generation over evolutionary timescales. What that means is that every individual is different at that level. And if we know that DNA is what's responsible for making protein, what type of protein, how much, when, what tissues, then if this is different among individuals, these proteins might be a little bit different. How much they express, how often they're expressed is going to be different. What tissues they're expressed in might be a little bit different. And that's going to be part of what makes humans different from each other. So that's what Dr. Silver is going to be talking about tonight. Dr. Silver got his PhD in biophysics from Harvard. He then went on to study both evolution and behavior in mice at Sloan Kettering and then later at Cold Spring Harbor. And so I had an interesting conversation with him when I picked him up from the airport, and I said, wow, you went from biophysics to bioethics. How did you make that jump? And Dr. Silver said, well, I don't think of myself as a bioethicist. I think of myself as an educator. What he seeks to do is to explain the biotechnology that exists and then couple it with what we know about how humans are so we can predict how that biotechnology is going to be applied to human nature. And he's been working to educate all of us on this type of relationship by doing a whole bunch of outreach, by writing a whole bunch of popular books, and then also writing articles in newspapers and magazines and appearing on TV programs. And here tonight for you, he will be talking about some of the very same things. So with that, I give you Dr. Lee Silver.
She wants you to do something?
Before I completely forget, we will have time for questions afterwards, so if you do have any questions, we're going to have a microphone up there. You can line up behind it. Thank you.
Thank you, Bryn, for that great introduction. And thanks for the couple of hours we had together. It's been really delightful. Thanks for the great weather here on the West Coast as well. Just going to get started. So, good. I like your colors. Great. I never saw a university before that had yellow as one of its colors. So let me give you a little bit of the background on where I came from. When I was in high school, I had a biology course where I was forced to memorize the Latin names for I don't know how many species and all of the names of the bones and the organs. And I vowed, that was in 10th grade, to never ever take a biology course again. And that lasted through college. I didn't take any biology courses in college. And it's probably a good thing I didn't, because when I was in college, none of the technology existed for doing anything useful, I would say. In biology. Biology was very descriptive, and that all just came about afterwards, and the science has been exploding ever since the 1970s, 1980s, 1990s, and the technology is becoming so powerful so quickly that I decided a couple of years ago that the best way that I could be involved in doing science was by stopping my lab, giving back my money to NIH, and sitting and doing science myself the way I like to do it. And we can all do that. So I'm going to talk to you today about the new biology, the new genetics, how it can be brought into our own lives and for fun. I'm not going to be talking about medicine. I'm going to be talking about just the excitement of learning about ourselves in ways that we never thought were possible before. And start with Watson and Crick, 1953, discovered the structure of DNA. And Crick, not only supposedly, but actually ran into the bar near where they discovered the structure and said to the other patrons at the bar, "I've discovered the secret of life." They probably thought he was crazy. But most molecular biologists would say, yeah, that was really pretty much part of the secret of life. And the secret of life they discovered is not always clear because it gets bogged down in molecular details. But the most important thing to understand is that genes are files of digital information, just like MP3 files that you can download from iTunes. And the genes are represented in a code that uses just 4 letters. We call them A, C, G, and T. And the information is in this specific ordering or sequence of those base letters along the DNA molecule. So this is why you and your sister or brother can share genes, even though, of course, you're not sharing any physical entities or physical material. It's all information. And in fact, the information in our cells, the information in all of our cells can be traced back, as we heard about the whole history of life, can trace back in a continuum back for over 3 billion years. That's the continuum of the information. It's remarkable because it's digital. It can be copied very faithfully. When I was younger, and I think when some of you were younger as well, instead of listening to music on iTunes, we had cassette tapes. And if you took a cassette tape and you made a copy of it to give your friend, and they made a copy, and they made a copy, and so on, by the time you had 100 copies, it was all noise because the information wasn't digital. It was analog, and you can never copy analog information accurately. Digital information you can copy accurately, and that's absolutely crucial. If the genes were not, there couldn't be genes that were not digital. So I just want to make it clear. And what Watson and Crick did is they could see this in the structure of DNA, and they could also see that the structure of DNA was redundant, so that the whole idea of the double helix, the reason that matters so much is because the 2 sides of the DNA have information that is essentially and metaphorically the mirror image. So you could divide the DNA up like this, and you can replicate the strand that's missing. So life can replicate. Just in looking at the structure of DNA, they understood how life can survive and replicate continuously over many, many generations. Now, in 1996, I wrote a book, and I got a lot of grief from scientists because they said I was speculating hyperbolically about the power of the science to come. This is 1996, 15 years ago, and among the many things I said we don't yet have the necessary technology, but it seems likely that the human genome will be deciphered by 2020. Turns out that I know I was right because the human genome was actually deciphered in 5 years after I wrote that. And everything else I wrote in that book about technology, everything that has happened since then has gone way, way beyond what I imagined. And I was being, people thought I was being hyperbolic. This is an example, and you'll see why I show you this. At the bottom is the total amount of DNA sequence that had been obtained. The government back in 1990 decided to put $3 billion into a project to obtain the DNA sequence of a human genome. It wasn't any person in particular. It was supposed to be the generic human genome. They put $3 billion, and they thought it would take 15 years to get there. And in 1996, we hadn't gotten very far, so I just sort of kept going with this line up in the exponential, and got to the year 2020, which is when I thought we would finish. What I didn't realize was going to happen is that the amount of DNA sequence we got would go up like that, and 5 years later, 2001, we had a complete sequence of the human genome. Cost $3 billion, as I'm going to show you in a moment. The cost has come down quite a bit, and the amount of time it takes has come down quite a bit. But I want to go back and show you a paper published by a graduate student at Harvard. It was very instrumental in getting me interested in this. It was the first seminar I went to as a graduate student, and it was a paper published by Nancy Maisel, who is now a professor at Yale. She had been a graduate student for 7 years, and her PhD thesis was 63 letters of sequence. That's what she got in 7 years. So she did 10 bases a year, basically, for 7 years. So that was the level of the technology when I got to graduate school. Then by the end of the 1990s, we had these machines. These are DNA sequencing machines, and you can see how big they are. There's a person up here in the front. This is actually a laboratory of Craig Venter, who is a biotech entrepreneur who challenged the US government to a race to sequence the human genome. And he would have won. Basically what he said is, I don't need any people, I just need a room full of robots and machines, and they'll work 24 hours a day and they won't complain about wages. And the government had finished 5%. He said he was going to do the whole thing in 3 years for 1/10 the price. He did. But the government, the leaders of the government effort decided they had to at least tie him in the race, because Venter had planned to sequence the whole human genome and then patent all the genes that he discovered. And Eric Lander and Francis Collins realized that if they could put the human genome into the public domain before Venter, then you can't patent it because it's in the public space, it's obvious. So in the end, Bill Clinton and Tony Blair had to make peace between the two, and they called it a tie, and the human genome's out there, and we all know it. What I want you to get a sense of is how quickly the technology is moving. These machines, they're big, right? 6 or 7 feet across. That was the state of the technology 10 years ago. And those machines then became miniaturized into microscopic dots. And so what you're looking at here is a picture under the microscope where each dot in this picture is performing the same kind of reaction that the machines were doing. So instead of having these giant machines that are spitting out DNA sequence, which was a lot faster than Nancy Maisel's, her 10 bases a year, you have all of these microscopic dots, millions of them, on a single little slide, and they're all doing DNA sequencing simultaneously, and it's all being looked at with a laser light. The little dots change colors. That's how they detect the DNA sequence. So that's an incredible level of miniaturization that occurred within a couple of years, and it keeps on going and going. I'll show you a slide of how quickly things are changing. And you might ask, well, we already have the sequence of the human genome. Why do we need more sequencing of the human genome? And the answer is because now we had a generic sequence, and we could go from asking what in our DNA makes us human to what in my DNA makes me me, and what in your DNA makes you you. By 2007, the human genetic variation was the breakthrough of the year. It sounds kind of boring because there wasn't a better term to describe it, but there was a breakthrough in 2007 when it became cheap and easy to look at the differences that exist between people genetically and try to correlate those differences with the different biologies that exist within people. So I want to give you a sense of how similar and different we are from each other. The human genome is 3 billion letters long. We actually have 2 copies of those 3 billion letters. And every person of the same sex is 99.95% identical to any other person in the room. I say the same sex because, as you all know, females have 2 X chromosomes, a male has X and Y. But if we ignore those sex chromosomes, everywhere else we're 99.95% the same. And it's like we have slightly different editions of the same book, and every once in a while there's a little change in one of the letters from one to another. Now, 99.95% the same sounds like it's almost identical, but this is digital information, so 0.05% of 3 billion is 1.5 million, and that's the number of differences that exist between any 2 people. I'm simplifying it a bit here. There's some other differences that exist that I'm not going to talk about, but basically this is the way to view the genome. We're 99.95% the same. There's a tiny little difference. So what we do when we want to study the differences between peoples, we ignore the 99.95%. That just makes us human. Okay, so we ignore that, and we just look at that tiny little bit that differs between us. 1.5 million what are called SNP differences, and that's what scientists have focused on for the last couple of years. There are DNA chips. Here's one here. You can see it being held in a person's hand. And on that DNA chip, you can look at 1 million SNPs simultaneously. You can take a drop of tissue, of DNA that comes out of a saliva sample or a cheek swab, put it onto that chip, and look at 1 million different places in the genome where people differ from each other, and then you can see how that person is more or less different at these points where people do differ. So that's a technology that led to a revolution starting a couple of years ago in the ability to look at individual genomes very cheaply and to generate large amounts of data, to compare people to each other. And this is a remarkable slide showing you how quickly technology is advancing. We all know about Moore's Law. Moore's Law is the black line here. This is a log curve. And so it says that computer technology, both the amount of storage on hard drives and how expensive the storage is increasing exponentially. And you see that as a straight line here on this chart. Meanwhile, DNA technology, the old technology, those big machines, that's the purple line there. And then starting in 2004, 2006, about that point in time, is what's called next-gen sequencing, where you take that giant machine and you miniaturize it down to a dot you can't see. And you see the red line there. That's the rate at which DNA sequencing technology is advancing in terms of how much it costs to sequence. And it's not just a question of cost, it's also a question of time. So the government spent $3 billion over 15 years to sequence one genome. And now you can do it right now. You could do it in a week or two for $10,000. So it's a huge, huge drop. And most molecular biologists think it's going to drop further even than that. It's very, very cheap to get DNA sequence information. Interpreting the information, that's what's difficult. So I'm going to talk to you now about how you or your children or grandchildren can go about hacking genetic information. So everything I'm going to talk about now is based on stuff that I have done since closing my lab. There's a company named 23andMe, and there's another company called DecodeMe. You send some of your spit to these companies, and they'll put it onto one of these chips and look at the million SNPs that you have. If you're doing it for health reasons, you're not going to get anything interesting, probably. That's not the reason to do it. If you're just interested scientifically, you're just curious about what's in your genome and where you came from, that's a reason to do it. And there are a lot of people out there who have learned how to read this DNA and take advantage of publicly available sources of data and programs to analyze this. So this is the page of 23andMe. That's me, actually. You can see I don't have alcohol flush. I don't taste bitter stuff. I have wet earwax. You know, it's kind of random stuff. And not very useful. Male pattern baldness, typical odds. Yeah, well, you know, it's. And I actually think the company is doing a very, very limited analysis. And they're looking at there's 1 million SNPs they're actually detecting in your genome, and they're interpreting a very tiny number. And one of the reasons for that is they're being challenged by the FDA and the AMA. There's a big battle going on right now about whether or not people should be allowed to look at their own genetic information. I'm not talking about me looking at yours, I'm talking about me looking at mine. And the AMA doesn't believe that should be allowed, basically. And so there were a number of companies that were providing this information, got scared out of the business. 23andMe is supported by Google money, so they have a lot of money to fight back against the FDA, which is what they're doing now. So the 23andMe, some of you may have actually done this kind of thing. And what you see on the website, I don't think it's very interesting. But what 23andMe allows you to do is to take that data, download it. That's what it looks like. Not very useful by itself, but there's these 1 million lines of SNPs, and each one of the lines over there, you can see where it says AGGG. This is the genome of a friend of mine. And there's another company called DecodeMe in Iceland. So they're out of reach of the FDA, and that's the same kind of information that they have over there. Those are the only two companies where you can actually get your DNA looked at without a doctor's prescription. So what can you see in your DNA if you look at it in this way? Well, one of the things is that because the DNA is digital, and because it's copied so well, there's only occasional mistakes that are made, and they occur at a certain frequency, at a certain rate over time. Your DNA is filled with whispers from the past that you can see. And to understand a little bit about our past, this is known as the human diaspora. Homo sapien evolved in Africa, in East Africa, until about 50,000 years ago. They were sequestered in Africa, and then starting 50,000 years ago, some groups ventured out into Europe and Asia and the New World, conquering the whole world and eliminating all the ecosystems as they went. But what happened because of that happened 50,000 years ago. People back then, they didn't have transportation. They couldn't get back to where they came from because, you know, they didn't have transportation like we have today. And so there were mutations, changes in SNPs that arose that we can now see patterns of human migration. And I'm going to give you some examples of these. This is one of the examples. So what you're looking at here, and this is all information, anybody can go to the web, go to the University of California, Santa Cruz website, and you can see this, and you can look at your SNP. Is there? Where's the? Here we are. Okay. Every SNP over here, this is the number of a particular SNP, and there's an A in some people, a G in others. The A is the original version, because we know that because the chimpanzee has the A. There was a mutation, the A changed to a G. And we can see that happened in the last 50,000 years because it's only present in Europeans and Asians. It's very prominent in East Asia. You look at, there's information like this on all 1 million SNPs, and you can look at each one of these SNPs and you can get a sense of when they occurred and what populations they're in. I think one of the interesting things about this, actually it shows it better on this slide here. So this is a SNP. What happened after populations went out into the Middle East is they split up, and some went to Europe, some went to Asia. This is a mutation that happened after that split, so you can see it's mostly in Europe. And one of the things you see on the edges of Africa are little bits of European DNA that are flowing back in along the edges there, probably because of colonial conquerors going in. And so you can see these little bits and pieces that give you some insight into possibly what's happening in people's pasts. Now, one of the things that's been very important in understanding how to interpret this is that our DNA is not inherited one base at a time. It's actually inherited as big blocks of DNA, blocks of chromosome. Whole blocks of chromosomes will be transmitted from one generation to the next. And so you can take your DNA, and what happens is that if you look at a particular block, this is a block of DNA, there are 5 versions of this block, and that make up 97% of Europeans. There are 4 versions of this block. That's 100% of Europeans looked at, 99%. So you see there's just a couple of versions that have survived in Europe of each of these blocks. And it means basically that the way in which human beings are unique is not because of any particular base, but because of the combination. So different people have different combinations. That's what makes us most different from each other, not the actual bases, which are shared. Interesting fact here is that this is a mutation over here from an A to a G, and that G is responsible for blue eyes. I was speaking a little bit earlier to some of the graduate students and commenting to them how we have a common ancestor from about 6,000 BC. I actually don't have blue eyes, I have brown eyes, but I have that mutation, and gave it to 2 out of 3 of my children. That mutation was derived, we are pretty sure, on the North Shore of the Black Sea, about 6,000 BC. So that's the kind of information that you can get from here. This is just looking at my own DNA. The red blocks are what I got from my father. The blue is what I got from my mother. And you can go along the whole genome and you can look at these blocks, and these blocks contain differences that are associated with particular traits and other kinds of information. This is kind of complex, so I'm just going to sort of pull out one little thing here, the kinds of stuff you can do. My own chromosome, looking at a particular chromosome, you look at the SNPs and you can actually see, I can actually see remnants of DNA from Africa, that's there in green, and remnants from East Asia in red, and the blue is European. So you actually can look along the DNA and see fragments that appear to be derived from different places. And I'll show you in a moment how this can go even further than this. You can look at, this is a friend of mine, looking at where she's inbred, where her DNA is identical on both chromosomes. This is, I made my wife to 3 different people, including self, who's on the bottom, my wife Sue, and looked at the DNA, looked at what the genomes would be like in the children she would have with these 3 different men. There's a way to interpret this that I'm not going to tell you right now, but it's the clearest actual children were when I mated her to Neanderthal. Because Neanderthal is so different in most regions of the chromosome than she is, that you actually can pretty well predict what the DNA is going to be in a child with Neanderthal man. She wasn't very happy about that. There's a mathematical analysis which I want to tell you about, because it's been very, very powerful in looking at the differences that exist, these tiny little SNP differences, and seeing really, really deeply into ancestry. And this analysis is called eigen analysis or principal component analysis, and it doesn't matter what it is or how it works. Basically, what you are able to see is you see pictures. Each one of these dots is a person, and the distance between dots represents the genetic distance between those people. One of the things that, and so I just did this on myself and a couple of friends. You can see my Euro-American friends over there, and my African-American friend over here, and then me and my wife and my father-in-law up there. What's going on? What's going on is that me and my wife and my father-in-law actually have an Eastern European Jewish heritage. And if you look at, and I gather you're going to have a whole talk about this in a couple of weeks, but this is an incredible result. Nobody expected this. That came out starting in 2008, and basically says that groups of people in Europe, this is, as you can see from the top, Irish, Germans, Spanish, Italians, Greeks, groups of people over the last 1,000 or 2,000 years have, in retrospect, it makes sense. And people didn't move very far from where their parents were. And so you get this, what's called genetic drift occurring, where populations actually look different from each other. The interesting thing about this line from Greece to Ireland is that it represents exactly the line of countries. It's the distance between the Irish and the Greeks is genetically and geographically similar. And then you see the Eastern European Jews over here, completely out of the rest of the European population. It means basically that for the last thousand years, they've been a very much inbreeding population, very distinct from others in Europe. What happens with populations like this, like the Eastern European Jews and the Amish, is they're a very closed community. So genes can flow out, but nobody goes in. Nobody becomes Amish, for example, even though Amish people who don't buy into the culture can leave. And so in Europe, the same thing happens, where nobody went into the Jewish ghettos. And you can actually do this thing here, actually, how Jewish are you? And over here, it's 4 grandparents is here, 3, 2, 1. It's pretty remarkable that you can see this. And the talk you're going to have in a couple of weeks and shows you this on a map. Again, this was completely unexpected. The fact that the distances between people map right onto Europe. The French, Spanish, Germans are up here. And the spots, so the little letters represent people. They were not put on top of the map. The map was put on top of the people. That's the distribution of the genes. It tells us a lot about what happened in Europe over the years. Very interesting is that the mountains, you can see how the mountains stopped migration between populations. So there's a new class of people studying genomes. The rise of the genome bloggers is the way Nature called it. A lot of people have put their genomes up on the web so anybody can download them and analyze them in relationship to each other. This is a story in December, just this past year, where somebody had put his genome on the web and he got an email, an anonymous email, once again telling him that he was part Jewish. And he said, that's ridiculous, must be something wrong with the data. And then he went and talked to his aunts and uncles and discovered, yes, there was this the Jewish ancestor. So the DNA told him something that he didn't know, and actually, even though he was a molecular biologist, completely disbelieved. A lot of research is actually going on with amateurs. And here's a website, Genomes Unzipped. You can see over here, I'm starting a public project with the aim of exploring genetic diversity around the world. There's more of this going on, I would say, among amateurs than among scientists, because this is just for fun. Scientists, if they do something, it has to be for, a reason that is coordinated with medical research that they're doing. And what's happening here in the underground is all of these studies that are comparing people to each other, getting a better idea. Anybody can do this, as I said, is you have a little programming experience, you know something about DNA, and if you're 16, it helps, as opposed to being 60. So on the web, there are websites where anybody can just go on, and you can use these professional tools, powerful, very, very fast tools to look at genomes and compare them to each other. One of the fun things you can do is take advantage of a discovery that was just made last year. There was actually a big argument among anthropologists as to whether human beings, those of us in the room, evolved simultaneously around the world from the more primitive human species, or whether, as I showed you earlier in a map, Homo sapien came out of Africa and wiped out, directly or indirectly, the other species in Europe and Asia. We know that Neanderthal man, from DNA analysis, we know that his ancestors entered Europe over 400,000 years ago, actually before Homo sapien became Homo sapien, and he survived until about 30,000 years ago, when Homo sapien, what we call modern humans, came and went through Europe. And it was assumed the debate was over. We went through Europe and Asia, we meaning Homo sapien, and the rest of the species are gone. That's my little diagram of that. But in fact, Neanderthal's not dead. A sequence of the Neanderthal genome was substantially completed. This is remarkable. This is what we thought was science fiction. Not 10 years ago, I'd say 5 years ago, we thought it was science fiction. The idea that you can take the tiny fragments of DNA present in the bones that are discovered 30,000 years old, sequence the genome of Neanderthal, and then amazingly what they discovered is that there were human beings who had fragments of Neanderthal DNA. They demonstrated that these fragments only occurred in Europeans and Asians and not in people who had pure African ancestry. So in other words, that's what you would expect, because Neanderthals were just in Europe and across a part of Asia. And so what this means is that, so the Neanderthal skeleton is on the left here, Homo sapien is on the right. Almost every person of European descent has some Neanderthal genome, and 7%, I think, is what the average amount of Neanderthal genome is in Europeans. And you can actually have fun with this. You can actually look at your DNA. Here's a little piece. Here's my own little piece of Neanderthal right here. And these are some other friends who have longer pieces of this. This is a region that's very common in Europeans with Neanderthal. So Neanderthal species did not go extinct. It survives within some of us, which is pretty remarkable. Probably wasn't very pleasant the way it got in, because the tribes of Neanderthals, and there were tribes of Homo sapien, and presumably the Neanderthals came and raped Homo sapien women, but the babies grew up and survived into us. So that's just kind of a fun thing to do. I now want to get into a topic that's, I'm sure, caused a lot of controversy, and that is the dissection of personality, human personality. And one of the things that I've been working with, my own research, is looking at how people respond to personality tests and dissecting the responses that are obtained with the sophisticated tools that geneticists use. Just one thing, this is just a picture of some of the genes that are known to affect personality, weakly in most cases. It's kind of one of the things you can do with your SNP data So studies have been done where they do brain scans on people with the two different forms of a dopamine receptor. And the form that I have on both of my chromosomes increases the activity in that part of the brain. So my brain wasn't scanned, but that's what I would expect if a brain scan was done. So let me talk about personality. One of the things I would say in my trip from physics down to biology, and every time I went down a step, physics is high, and then you go down, and biochemistry is lower, and then you go down and down and down. Everybody's always saying how you're taking the wrong path. So when I went from physics to biophysics, my physics professors were very upset with me. So finally, I've gone all the way down to psychology, and I guess you can go further, but. And in psychology, psychologists for many years, actually all the way back to the Greeks, have been trying to understand the variation in personality that exists among people. And I attempted to understand what they understood. Basically what the assumption was is that we can break down personality into a very small number of types, and you can list them, and people fit into these different types. And there are some theorists say there are 4 types. That's what you see there on the left, and then there's 6, and then there's 8 and 16. It's always a symmetrical kind of thing. You may have Myers-Briggs. That's what this thing is down here. It's very popular. Lots of people have been given this test. And then they pigeonhole you into one of these categories, and that describes who you are. Myers-Briggs was actually created by a woman without any scientific training back in the early part of the 20th century, who noticed that her son-in-law was different from others. And you can look it up on Wikipedia. Look up Myers-Briggs. You'll see. And that was the basis for the test. And she read about the personality types that Aristotle had defined and Carl Jung, and that's where most of these things came from. So I was very lucky because I'm going to just for a second. I became very friendly with an anthropologist named Helen Fisher, and here she is. Helen Fisher works with a website named chemistry.com. It doesn't belong to the American Chemical Society. It belongs to a dating website. They convinced Helen to work with them. She designed a questionnaire, which is, she also said there were 4 personality types, very, very simply. But she has gotten 8 million responses to this questionnaire. People fill out this questionnaire, and then she'll tell you who you're more likely to match up with romantically. She has evidence that there's a slight difference in terms of how you will match up. So if we go back to the slide, what I decided to do with the 100,000 responses initially was to use the same kind of analysis that geneticists use to track differences between people. And what I did here is these are not people. These spots on these graphs are the ways in which people answer questions on this personality test. What you see here, so these are, for example, 4 kinds of questions that in this particular area of personality space are very highly correlated in the way that people answer these questions. And these are questions that look at inner emotionality. This is empathy-like questions, cautious and conventional. These are sort of the personality types, the broad personality types that have been described. I was interested in seeing whether that was all there was, or was it deeper, and discovered that indeed it was deeper. that if you go deeply into all of these questions, you perform eigenanalysis, what you see is that there aren't just 4 or 5 personalities. There are many, many dimensions in which there are reproducible correlations. So what I'm referring to as personality components, where out of 100,000 people, there will be 500 who are very, very similar to each other. And if you look at another 100,000 people from North America, this is North America, you'll find again 500 people who fit a particular set, answers to a particular set of questions that are totally bizarre. But there they are. Those 500 people answer those questions in the same way. This is at sort of like the high level. You can see this. I'm going to go past that. You can actually put people. If you see the bullseyes on this slide here, where people fit in particular dimensions. And this was kind of fun, telling people what they're like here. This is a friend of mine, Anne. And she's positively drawn to these traits and negatively drawn to those traits over here. She's not cautious or conventional. She in many ways, and she's not rational or assertive, which she agrees with. But that's, and I should say that this is me over here, overlapping with her in some ways. But what was more interesting to me, I have a pretty simple personality and just sort of straightforward novelty seeking. That's what I do. Was most interesting is that most people are not like that. Most people, if you look at their responses, the negatives here are in red all over the place. Positives are in green. And so they don't fit into all the personality models that psychologists have been putting forward over the years, because this person here looks like he's a combination of everything, whereas yes and no. It doesn't seem to make any sense in this particular plane. But when you go deeper, which is what you can do with the eigenanalysis, you look at him over here, and these are dimensions where there's a small number of people, as I said, out of 100,000. In this case, there's 700 who have this particular combination of traits that you would not expect to find randomly. So that's, and this is another person here who has another set of traits, also negative against some traits and positive against some others. Let me go past this. These are just slides to say that there are some complex traits that have been looked at in detail, and we pretty much have a good way of working things out for hair color, for example. This is eye color. This is all being done by forensic scientists, because you don't need to do a genetic analysis on yourself to figure out what your hair color is. Your spouse might want to know if you're coloring your hair, but that's different. So forensic scientists like to take, you take a drop of blood left behind at a crime scene, you look at the DNA, and you can figure out hair color, eye color of the person who committed the crime, and many other characteristics. I mean, all the physical characteristics are going to come out, and that's why these kinds of studies are being done. But one of the ways in which you can have fun with this is by putting together people's genomes, as I've begun to do, and then figure out whether two people are going to have a kid with different eyes or hair or any of these kinds of traits, which is kind of fun. Going back to my wife, here she is mating with several different people, and I could tell that it's very interesting with certain people, all of her children would have blue eyes. With other people, there was a 100% chance of brown eyes. And with me, it was 50-50. And I have 2 kids with blue eyes and 1 with brown, which is close as you get to 50-50. So I hope to just have shown you that there's fun we can have with this kind of stuff, that you can use genetic understanding, I think, to learn more about yourself, learn more about other people, learn more about how you are similar, how you differ from other people. And it's something that is the information, the programs, it's all just sitting there on the web, public access, and anybody can get into it. It's because of the way the Human Genome Project was done, which is that all the data goes out. You have these huge data sets anybody can see, and that's the reason this technology has been moving forward so quickly. And I will stop there and take questions.
How much of us is DNA? You have to sequence to identify this history of that person with Osama bin Laden.
So one of the question is how much of us is DNA? And I'll tell you a little story about insurance discrimination to give you a sense of the answer. I don't know about Oregon, but in New Jersey, if you have a particular gene and you're between the ages of 18 and 25, your insurance costs, car insurance, is $500 more per year than if you don't have the gene. The gene is called SRY, which is a gene on the Y chromosome that causes men to develop as men. So the point to make is that for the insurance companies, this is fine. Boys between the ages of 18 and 25 have more accidents as a population than girls of that age. So that's what the insurance companies will say. But you cannot use the fact that somebody's a boy or a girl to predict whether they're going to have an accident or not. It's a population level difference in the average rate of having an accident because you're driving crazily. When I was looking at the personalities, it was most clear at the population level, not at the level of individuals. I could put individuals onto this map, but it became much, much clearer when I could look at the whole population, where you could begin to see the different kinds of personalities that exist. So how much is genetic and how much is non-genetic? It's impossible to answer that question because it's going to differ. I mean, you don't when you have an answer to that, that's not at the individual level. That's at the population level.
Osama bin Laden was identified by his DNA.
I think Osama bin Laden was identified by his face, actually, right? They took a picture of him, sent it back to the CIA, and it's a 95% probability that it was the same person as the man who was seen before. I don't know if they look at his DNA.
There was some
They did DNA fingerprinting.
His sister died in Britain, and her brain was confiscated, and they were using that for comparison.
So they looked at Osama's
So how
DNA.
much comparison is there between his sister's brain and his?
So anybody know whether they've actually done an analysis on the body?
Pretend that whatever they did with his sister's brain,
Yeah.
they did with Osama.
And they got results.
But how sure?
Yeah. Okay.
So if they did that, how sure would they be that it's him
That it's him as opposed to his brother?
or a stranger?
I don't know. I haven't seen it, but I don't know how they could. They're looking at his sister.
So they're comparing his DNA to his sister's.
Well, then it could be him or his brother. I mean, so siblings will have a certain. They'll be able to determine that it's a sibling of a sister, it's a half sister, I assume, which would mean that you couldn't distinguish between Osama bin Laden and any of his half brothers.
Just bring up another question that came up in the last lecture about the influence of politics or DNA in our political sense. But rather than that being the front of my question, I'm wondering about whether you can talk about epigenetics. It said that the mapping the human genome was going to be very
So what's your definition of epigenetic?
My understanding from the little reading I've done about it is that it depends on where the methyl markers are on any particular gene. So whether those methyl markers are turning on the action of the gene or not turning it on, and how those might interact with one another, other methyl markers with other genes. There seems to be some evidence, for instance, that obesity can be inherited from 2 generations back, but still a generation in between, depending on environmental influences. And it's actually the environmental influences that seem to be most interesting for those methyl markers being activated or not.
So this is a more of a political question than a scientific one. When I wrote, I have a textbook in genetics that I co-authored, and I was assigned epigenetic as one of the words in the glossary. Epigenetic, initially, when it was first used, it meant anything outside the gene, which meant the proteins, the RNA, any way in which the organism differed that wasn't genetic. So we've known about that for a long time. When molecular biologists talk about epigenetic now, they're often referring to this chemical modification of DNA. Most of the time, those chemical modifications are identical between people. Most of the methylation that occurs is identical. I think there's that this term epigenetic is being used in ways that are not necessarily scientific. And the idea of skipping a generation is something that people assumed until Mendel. You saw things in one generation, you didn't see it in the next, and then you saw it in the following generation, which made absolutely no sense before Mendel's genetics. But then once Mendel gave a genetic explanation for inheritance, it no longer was seen as skipping generations. I don't know about the particular data you're mentioning here. I know that there have been a number. There have been some findings. I mean, there are very, very small differences that have been found. The question is whether they're reproducible or not. I don't know how much that they will contribute to the question.
Given the speed with which science has evolved in this area, I would imagine within 10 years or less, people will easily be able to identify specific traits associated with their children if they decide to get their genomes investigated.
So you mean before they conceive?
Right.
Right. So what part of my research is to look at that very, very question. And the answer is it's probabilistic and it's different for any couple. So that I look at myself and my wife, there are some regions of the genome where I know absolutely what's going to be inherited. Other regions of the genome where I know a little bit more than what would happen randomly. Other regions of the genome where it's no different than random.
Do we have any research thus far to indicate a propensity to violence as opposed to a propensity to peace or any other morally questionable activities? Do we have any sort of indication thus far?
Well, the first thing is that people have to define those words. Those are very. I mean, when I did behavioral genetics in mice, it was very, very difficult to actually find anything. I can tell you that any attempts to look at the genetics of personality have all failed, and which is not unexpected because you have these very broad concepts like you're saying, violent, not violent. For a geneticist, though, that is much too broad a concept to investigate genetically. And when you're talking about terms like that, I mean, it's obviously very clear that it's not just the genetics.
And the other question I have would be, since
99.95.
95% of our traits are similar, are we focusing at this point on that 0.5% that are different?
Yes. So I would say the whole point of the Human Genome Project was to understand how human beings differed from other forms of life, and that's looking at the 99.95% that we all share, and saying how are humans different from chimps. That's what everybody wants to understand, or from any other kind of animal. When we're focusing on the 0.05%, we're talking about contributions to human variation, usually complex contributions. I mean, it's very easy to go look at the DNA and go and look at your ancestry. That's fun. It's digital, easy to do. Learn about yourself, not so easy. I mean, most of the stuff is probabilistic. But the whole point of concentrating on the differences is so that we can understand variation. If you look at the people in this room, they all look different from each other. Why do they look different from each other? Well, they're genetic differences that, as we know, are most responsible for physical differences. And there is variation, so we want to understand the variation. That's why you ignore the generic human. Not that we know it completely, but there's a difference between looking at the variation versus the species as a whole definition.
With retroviruses like AIDS, for example, would it be possible, like, that the effects of a retrovirus by genetically engineering a retrovirus that could reverse the effects of the original retrovirus?
So retroviruses are actually very interesting because they can hide out inside your cells not as viruses but as virtual viruses. In other words, they copy themselves into the chromosome, which is why it's so difficult to attack them, because they don't exist. All they are is information. So it's one of those things which is absolutely fascinating, which is here the retrovirus has converted itself into pure information hiding in your DNA. And then with HIV, for example, at some point the information that expresses itself into viruses. But that's why, for a long time you can't even see it because it's hiding. It doesn't even exist. So this whole idea of information is very interesting. It actually goes further than just the genes. If you look at who you are a year ago, most of the physical material that makes up individuals is turned over very quickly. So physically, you're not the same as you were a year ago. But, psychologically you are. It's the information that has continued. So anyway, that's the retrovirus story.
How much of the genome, as we go back and study it forensically, where's the threshold of transition between plants and animals, bacteria and plants, and so on? How much of it in terms of the whole entire
Somebody else could probably give me plants and animals are billion years apart or something like that.
Are there still vestigial?
Oh, yeah. Are there yes. You could actually see, so the question is, I'm sorry, I didn't understand the question. The question is, can you actually see the information in us and other living things, and not just plants? You can go all the way back, and you can see the remnants, and you can see the information there, the information in our own cells, and you can see the similarity. In some cases, some genes are very highly conserved, which could not happen by accident. It's the strongest evidence that there is, that exists, that we all diverge from a common life form a long time ago.
Okay, well, if we could thank Dr. Silver one more time. This is an incredibly shameless plug, but if you like what you saw and you want us to do more public outreach, we are accepting donations in the lobby.